Welcome to Click for Biology. We are considering genetic modification, and here we're looking at the genetic engineering of a useful product called humulin or human insulin. The problem that we are trying to solve through this genetic engineering is that ordinarily people have a gene in their pancreatic cells which expresses the protein called insulin, and that is a hormone. This hormone is used to regulate blood sugar. There are some individuals with an autoimmune disease that destroys the beta cells of the pancreas, and they are unable to manufacture the insulin. These people are suffering from a condition called type 1 diabetes. They cannot regulate their blood sugar. And therefore, to treat people with type 1 diabetes, it's necessary to obtain a source of human insulin, and this is today achieved through genetic engineering. In genetic engineering, we have some tools for the production, and these include restriction enzymes, an enzyme which will cut through the DNA at a particular location. There are a wide variety of such enzymes, and they cut DNA at different positions. These are called restriction enzymes. Working in the opposite direction, we have DNA ligase. DNA ligase takes fragments of DNA and joins them together into the single structure of DNA. So working in opposite directions in their action. These two tools are essential for genetic engineering. In the production of this genetic engineering, we are going to manipulate DNA and then use this with bacterial cells for the actual production. We begin with some bacterial plasmids. These are small circularized regions of DNA which bacteria normally carry, and we will cut into that with a particular restriction enzyme. Let's say that it cuts it at the base sequence ATA. We would then use the same enzyme, the same restriction enzyme, and cut into the human chromosome. So this would be the human chromosome, and we are going to cut at the location of where the gene for insulin sits using the same restriction enzyme. And here's the human gene here for insulin. We then combine the bacterial DNA plasmid with the human insulin in the manner shown in the diagram here. And of course, we are able to join the DNA together using the enzyme DNA ligase. The combination of the bacterial plasmid and the human chromosome is what we call recombinant DNA, recombined DNA. Taking this recombined DNA, we can move towards the production of insulin itself, and to do so, we have to transfer the recombinant DNA to the bacterial cells of the species which is normally E. coli. This is achieved through a number of different means, including the use of calcium chloride, and also a voltage can be used in order that the plasmid will enter the cell. And so then we end up with the bacterial cell which contains the recombinant DNA, the bacterial DNA, and the human insulin DNA, and this type of cell is then known as a bacterial recombinant, or one could call it recombinant E. coli. This gets us to the position where we can begin the production of the actual human protein. There is an alternative pathway to be mentioned, and that is that with the insulin gene cut from source, it can be introduced into the viral capsid. This is the shell, the protein shell of a virus, and we can make a recombinant in that way. These viruses are then introduced to the E. coli cells, and because they are bacteriophages, viruses of bacteria, they are able to introduce the copies of the gene into the bacterial cell in this manner. And again, we end up with a bacterial recombinant. Now we need to move forward to the actual production of the human insulin, and this is achieved using a structure called a fermenter. The fermenter will begin with introducing the culture of transformed bacterial cells, the inoculum, into the fermenter itself. And there we want to create the optimal growth conditions for the bacteria so that the population of bacterial cells increases. So the theory here is that the bacteria will actually begin to switch on the gene, express the human gene for insulin, and they will manufacture that particular protein. To achieve this, we have to establish the optimal growth conditions, and one of the factors to be regulated is temperature, and another one would be pH. Temperature is often regulated using water jackets or water coolers around the outside of the fermenter, and by regulating the temperature through a probe, we're able to control the internal temperature through regulating the water flow. Gases also need to be regulated, and we have an air inlet and an air outlet in order to achieve that. The progress of the reaction is monitored by taking regular samples. The bacterial cells in the inoculum are provided with nutrient, and of course an important part of that will be a source of energy in the form of carbohydrates, but also amino acids appropriate to the synthesis of insulin. When the production is complete, this is drained from the fermenter, and we would then end up with a mixture of the bacterial cells, the various components provided to the bacterial cells, and of course the product itself. And then begins a long process of downstream regulation in which there is purification of the insulin product so that it is in a form which can be then injected into humans. And this one is called Humulin, human insulin.